If you're watching this video, we have about 10 minutes before we start. Before you skip ahead 10 minutes, I'll remind you, because you're watching the video, in addition to sending the attendance words, you also need to send a screenshot of when I say the attendance words. As usual, make sure the screenshot shows your entire screen. So for example, I can see what time you were watching that video. So if I needed to, I could cross-reference my YouTube account and, make sure that someone was actually watching it at that time and you're the only person who was watching it at that time, so on and so forth. But anyway, that's the only reminder. And I guess your first word for attendance, so you'll need to take a screenshot for this, will be empty. Because right now this classroom is empty. So your first word for attendance is empty. Now you can skip ahead about 9 minutes if you want. And then once everybody else gets here, I'll start their attendance words. But yes, you have an additional attendance word. Yeah, pen I can borrow.
I personally don't.
Nope, I'm sorry. All right, now that we have 3 people here, I'm gonna do an extra credit word. So this is unofficial, since we haven't started yet. But the first unofficial word for attendance is going to be marker. So if you guys decide to send the words in, you get extra points. John, when you send the words in, if you also send that word marker, then you get more extra credit points for everybody else, because technically you 3 are the early birds with 1 minute left to go. All right. That being said, speaking of only having 1 minute left to go, let me do some attendance. If you're online and I say you're not here, don't worry about it as usual. I'm just talking about the people who are physically here. Bangura is not here. Case Bolt is here. Tristan is not here. Covington Griggs is not here. Kately is not here. Dow is not here. Hughes is not here. Lebdi is here. Carla is not here. Mankins is not here. Mathis is not here. Rains is not here. Reed is not here. Renee is not here, or Elise. Thompson is not here. Withrow is not here. Yindui is not here. Elkins is not here. I would ask if I missed anybody in person, but there's only 2 of you, so I know I didn't miss anybody. It's 8 o'clock. We'll get started. This is going to go the way it usually does. If you're in person, you can send me the words if you want to get even more extra credit, but you don't have to because you're physically here, so you're marked as present. If you're online, you have to send the words to get marked as present, just as usual. That being said, let's jump into it. I'm not going to do the announcements yet. I want to give some more time for people to show up. This is where we left off. We already talked about prophase 1. Actually, no, we didn't. We didn't talk about prophase 1. I wanted to, wanted to start there. That's a good place to start. So here we go. We were talking about prophase 1. So there's the first word for attendance. What are we talking about first? Prophase 1, metaphase 1, anaphase 1, or telophase 1? What is this lecture starting with? Nice and easy, right? Anyway, actually, even before I start talking about it, I'll remind you, as far as the exam is concerned, mitosis, the questions on the about mitosis are very going to be much like, what stage does this happen? What stage does that happen? What are the correct orders of these stages, so on and so forth. With meiosis, it's going to be a little bit different. A lot of the questions about meiosis are going to be comparing and contrasting how meiosis is different than mitosis. And then there's going to be some comparing and contrasting between how meiosis 1 is different than meiosis 2. So keep that in the back of your mind as I'm teaching you this. Also, I'll remind you, in my opinion, the best way to learn this is to make sure you're very familiar with mitosis and then start learning about meiosis, right? Because if you think about it, if you're familiar with mitosis, then when you learn meiosis, you could almost think of it like as a variation of mitosis. So that being said, let's talk about that. So prophase 1 is basically like prophase of mitosis with some exceptions, and you need to know these exceptions. Once again, if we're dealing with any of meiosis 1, right, whether it be prophase, metaphase, anaphase, or telophase, if it's meiosis 1 that we're talking about, we are dealing with homologous chromosomes, not sister chromatids. Right? We're not even concerned with the sister chromatids yet. And to remind you, mitosis was all about the sister chromatids. We never even mentioned homologous chromosomes in mitosis. So if there's any questions about homologous chromosomes on this exam, for the most part, at least in the context of mitosis and meiosis, you should associate it with meiosis. Never mitosis. Now, if you want to get even more specific, which you do need to get more specific for the exam, now I can tell you again that if we're talking about homologous chromosomes, we are also specifically talking about the first round of meiosis, meiosis 1, not meiosis 2. So these homologous chromosomes, again, they paired up because we already talked about how they have crossing over because they're in such close vicinity. That pairing is called a synapsis. I'm going to put a question mark next to this, meaning I don't know if I'm going to ask you questions about that or not. That's definitely a word you should understand, though. That way, when you're studying this process and you're reading your textbook, you know what it means. So you can almost think of it like, remember when we talked about chromosomes? Like, you got this one chromosome right here. It's a horrible drawing, right? That's a chromosome. Let's throw it back to mitosis. That's a regular old chromosome, and it's a G1, right, of interphase. Now it goes through S phase of interphase, right? And what is S phase? That's when the chromosome duplicates itself. So now, instead of having one chromosome, you have two sister chromatids, right? And they are attached right here at the centromere. So we talk about how these things are attached. So now, kind of in the same way, so here's one homologous chromosome, right? So we're done with this one over here on the left. Here's another homologous chromosome. So now we're done talking about sister chromatids. We're talking about homologous chromosomes. And when those things are together, we call that a synapsis. There you go. Now you know. Again, like we mentioned earlier in the previous slide on Wednesday, prophase 1 is when crossing over occurs. Crossing over is going to be very important when we start talking about genetics. So this is not trivial information. Not only will it be on the exam, but it'll be important for understanding things later in the semester. And again, if you read the textbook, your textbook actually goes into more detail and splits it up between prophase and prometaphase. I'm keeping it simple. So for the purposes of the exam, you can combine those two. Prophase and prometaphase, as far as we're concerned, for this 100 level non-biology major class is just prophase. Any questions about this slide before I show you some pictures? Actually, this next slide is a really bad picture because it's all the pictures, but we'll go ahead and look at it. So here's this is really, I need to fix my PowerPoint. Not only is this all the pictures, which I'm not ready to show you yet, but it's also meiosis 2, which we're not even there yet. So forget that picture. Pretend like you didn't see it. Also, that picture's not from your textbook, just so you know. Anyway, so what comes after prophase 1? Metaphase 1, right? It's always prophase, metaphase, anaphase, telophase. Again, this is essentially the same as mitosis, except, once again, it's not the sister chromatids that we're talking about meeting in the middle, it's the homologous chromosomes. And once again, you should associate homologous chromosomes with meiosis, not mitosis. And then to get even more specific, once again, you should associate homologous chromosomes with the first round of meiosis, not the second round. Now, here's where it's going to also get very important later in the semester. When these homologous chromosomes line up in the middle of the cell, it is random. So it is not as if, remember, like I said, for homologous chromosomes, you've got 23 pairs of chromosomes, 23 pairs of homologous chromosomes. For each one of those, you have a mom, you got one from your mom and one that you got from your dad, right? So if you look at chromosome number 17, for example, one of them is from your mom, one of them is from your dad. Now, they're all going to line up. All 23 pairs of them are going to line up in the middle in metaphase 1, but it's random. And what I mean by that is it's not like, normally I draw the chromosomes, but in this case, I'm just going to use letters. It's not like you got the mom chromosomes on this side. Oh, that's so hard to draw. And the dad chromosomes on this side, right? It doesn't work like that. I mean, I guess it could, especially in this case when we're only dealing with 3 pairs of chromosomes. But it's not like that, right? It's completely random how they line up. So think about that. 23 different pairs. Think about all the different ways they could line up with the mom on one side and the dad on the other. That is important. That is something called independent assortment. There will be questions about that on the exam. When we get to the next chapter, we're going to talk a lot more about independent assortment. This is just your first introduction to the topic. Independent assortment. It's a very important concept. And hopefully when I give you these numbers, because of independent assortment, like the chances of you being you, numerically, mathematically, are mind-blowing. Like there's no other way you could ever, there's no other way that you would ever be created. Right? You are completely unique, and we'll talk about that later. But anyway, yes, that is independent assortment, at least for now. So you definitely need to know that. Independent assortment means that when those homologous chromosomes line up in the middle of metaphase 1, it's random the way they meet up. It's not all mom on one side and dad on the other. It's completely random. So, the next word for attendance. For metaphase 1, when the homologous chromosomes meet up in the middle of the cell, is it all mom chromosomes on one side and dad chromosomes on the other? Or is it random?
Can you say it again, please?
Huh?
Can you say it again, please?
Sure. So, the next word for attendance, and I don't normally do this, the next word for attendance, the one I just asked, is when the homologous chromosomes line up in the middle of the cell for metaphase 1, is it all mom chromosomes on one side and dad chromosomes on the other? Or is it random? And that's it for metaphase 1. Are there any questions about metaphase 1 before I show you another slide that doesn't belong in that spot? Okay, so again, we're skipping this. I don't know. My bad. These are meiosis 2 pictures. We don't need to worry about that. All right. Now let's talk about anaphase 1. And even though it's not put there, telophase 1, right? These are the last 2 stages of the first round of meiosis. Once again, it's essentially the same as meiosis, except, once again, we're dealing with homologous chromosomes. We're not even talking about sister chromatids yet. So once again, you should associate homologous chromosomes with meiosis, not mitosis. Once again, more specifically, you should associate homologous chromosomes with meiosis 1 and not meiosis 2. But yeah, it's essentially the same. You should remember in anaphase, in anaphase and mitosis, the sister chromatids separated and they were moving towards opposite ends of the cell. In this case, it's the same thing, except it's the homologous chromosomes that have separated and moved to the opposite ends of the cell. In telophase, remember, in telophase, actually, I'm going to leave this up for independent work because I don't think your textbook gets into it. Remember, in mitosis, telophase kind of ends the process, other than cytokinesis, right? Cytokinesis also splits the cell. But in telophase, as far as we're talking about chromosomes are concerned, that was the end of the story for the chromosomes. Everything was in reverse, right? So the chromosomes uncoiled, the mitotic spindle disappeared, The nuclear envelope reformed. That was the end of mitosis, right? That's how it ended. That's how telophase ended in mitosis. So my question for you for independent work, because I'm not going to give you this information, and you don't need to know it for the exam. What happens in telophase 1? Because think about it. So sure, we're done, but now these cells are going to have to split yet again. And now we're going to have to separate these sister chromatids. So we're not done yet. So logically, I don't know, like, does the nuclear envelope reform? Do those chromosomes uncoil? Does the mitotic spindle disappear? All those things that happen in the end of mitosis, do they happen in the end of meiosis 1? Obviously, they do happen at the end of meiosis 2, even though we haven't gotten there yet. That's obvious. So my question for you for independent work, if you want to look into it, what exactly does happen at the end of meiosis 1? Of course, it's been a while since I looked at this new textbook, so if it's in there, that might be your answer. Anyway, any questions about this slide? We're still going to talk about, any questions about this slide or meiosis 1 in general? We are now done talking about meiosis 1. Okay, and again, hopefully you picked up on this. For the most part, think about what we've talked about. For the most part, the most important thing, the most repetitive thing that I've said in this discussion is that for every one of these stages of meiosis 1, it's all been about homologous chromosomes as opposed to sister chromatids. So hopefully you picked up on that pattern, because that's largely what the questions are going to be like on the exam. All right, so that brings us to meiosis 2. This is essentially the same. This is more than, so for both of them, I did say, hey, this is essentially the same as mitosis, except, and I'm going to do the same for meiosis 2. But if there were a test question that said, which one is more like mitosis? Meiosis 1 or meiosis 2? The answer would be meiosis 2. So let's talk about some of these differences, though. At the end of meiosis 2, which is, the end of the whole process, the end products are cells with half the number of, or half the genetic material. So you definitely need to know that. Remember, in mitosis, you start with 1 diploid cell, you end with 2 diploid cells. In all of meiosis, when you combine meiosis 1 and meiosis 2, you start with 1 diploid cell, you end with 4 haploid cells, right? So we cut the genetic material in half. That should be easy to remember, because remember, the biggest thing you need to remember about meiosis is that the purpose is to create sperm and egg. And if you can remember that, then surely you can remember a sperm and an egg is going to need half the genetic material because it's going to combine. There's no other way for it to work. You can't combine two full cells, right? Because then you'd have double the genetic material. And that's not good, right? So again, the end products are haploid cells. That would definitely be a test question. Which one ends with haploid cells? Meiosis 2, or meiosis or mitosis, right? And the answer is meiosis. Of course, you probably already know that from the pre-lab that you did from last week. And again, like I just said, here it is again in writing. Mitosis, you end with 2 cells. Meiosis, you end with 4 cells. In mitosis, those 2 new cells are identical to each other, and they're identical to the original cell. Here in meiosis, all 4 of those cells are genetically unique. Unique from each other, unique from the parent cell, completely unique. Any questions about this slide? All right, I wish I had pictures. Apparently, I just lost the pictures of meiosis 1. But here it is, meiosis 2 again. So again, this is mostly just like mitosis, because here is what we're dealing with. Like the homologous chromosomes have already been separated, right? Because this is over here, you can't see it for prophase 1, right? What we did was we started with 1 cell, and then eventually it's separated into those 2 cells you see right there, right? That's meiosis 1. And I'm sorry if you're online, you probably can't see that, but you're not missing much, trust me. Anyway, so now these 2 cells, it's just like normal, right? So the mitotic spindle is formed. It's going to attach, which they haven't shown them attached to the sister chromatids yet, but it's going to. The nuclear envelope has disappeared. And again, I'll remind you for independent work, I'm asking you, did this happen between meiosis 1 and meiosis 2? Or did it just never, did all these things just never disappear at the end of meiosis 1? We looked that up for independent work. Anyway, These things get attached as usual, then they start moving towards the middle of the cell, which is not being shown. Once they get to the middle of the cell, that's metaphase. And again, this looks like mitosis, right? Now we're dealing with these sister chromatids that are lined up in the middle of the cell, not the homologous chromosomes. We've already separated them. Granted, the picture's a little bit different because you can see these chromosomes have multicolors, which is just to remind you that crossing over happened, right? So this is a mom. Let's say that's a dad chromosome with a chunk of mom on it, and that's a mom chromosome with 2 chunks of dad on it, right? So on and so forth. And that's metaphase. And what you can't see in this picture is that these mitotic spindles are trying to pull these sister chromatids apart. And then finally, once they are separated, that's what we call anaphase, right? And what you cannot see in this picture is that now that they've been separated, they're now moving towards opposite ends of the cell. The cell itself, even though you can't see it, is going from a circle to more of an oval, right? It's becoming elongated. And then finally, we have telophase 2, where the whole process is done. And then cytokinesis, right? So it splits those cells. So now we have 4 daughter cells. So again, meiosis 2, as far as we're concerned in this conversation, at this level of academia, meiosis 2 is basically the same as mitosis, right? Because we're dealing with sister chromatids. We're separating sister chromatids. But again, one of the big differences that you can see on this slide are we have 4 cells and not 2. And they're haploid, they're not diploid. And they're genetically unique, not genetically identical. So that is it for the details of meiosis. The next section we're going to talk about is kind of a review. It's going to be like, all right, now that you know this, it's kind of opposite how we've done it before. It used to be like big picture, and then we get into the details. Now it's like we've already done the details. Let's take another step back and look at the big picture to make sure you understand the big picture. And again, my recommendation for studying, because most people's brains that I've encountered work like this, learn the big picture stuff first before you start adding details. If you can tell me all about prophase 2, metaphase 2, anaphase 2, and telophase 2, but forget the fact that the purpose of meiosis is to produce sperm and egg, then who cares? Like, who cares about all these details if you don't remember the most important parts of it? So anyway, yes, the next section we're going to talk about is going to be a review. I'll remind you, this bullet point is mostly based off of my old textbook. So if you're following along in your textbook, the new one, it's going to be a little bit out of order. And also, obviously, some of the pictures are going to be different. Now let's jump into announcements and reminders. It's almost 8:20, so even late people should be here by now. I'll remind you that spring break starts today, right? So you've got all next week off. You should use that time wisely. So if you need a break, take a break. If you need to get caught up with some work, get caught up with some work. You guys are on spring break. Technically I am, but I'm going to be working. So if you need me, if you need me next week, now's the time to do it. A lot of you need catching up. I'm here to help. Lab 7 and pre-lab 8 are due today. As usual, I recommend that you focus on the pre-lab. This pre-lab is important. Last week's lab, or this week, technically, lab 7, as you know, was virtual. When we come back the week after spring break, that lab 8, is not virtual. We're going to be doing it as a team. So you need to, this is more important as usual, for you to be prepared. Because if you're not, then all your classmates are going to suffer. Let me see if I can click this and show you something really quickly. Yes, all right, let's take a very quick look. So people have already messed it up, but it's not due yet, so I applaud you for getting started early. Oh, come on. Really? Not going to show me? What? Well, maybe not. Never mind, I was going to show you pre-lab 8. So I'll try to explain it verbally. Pre-lab 8, you have a video with a set of questions, like you sometimes do. Then you have an introduction you need to read. You need to actually click a link, it'll bring you to the introduction. Then you have pre-lab questions, as usual, to prove that you read that introduction. Pay attention, this is where it gets a little bit different. Then you have a set of hypotheses, right? So it's going to be like, hey, do you think there's going to be more people in the class with a cleft chin or without a cleft chin? Do you think there's going to be more people in the class with blue eyes or non-blue eyes? It's those kind of questions, right? Those would be the hypothesis questions. Now pay attention, this is where people have already screwed it up. I shouldn't say screwed it up because maybe they just haven't finished it yet. Keep going. There's 2 tables. You need to fill those 2 tables out. So for example, it's going to ask you what your trait is and your genotype. So for example, just using the same example, if you have blue eyes, you'll put blue in one of those things. On one box, you'll put, well, blue eyes is recessive, right? So hold on. In that table, there's a few things you could write. You could write blue, right? If you have blue eyes. Oh man, this is so hard to write on. Ridiculous. Anyway, you could write maybe type out the word blue, which I'm not going to do because this thing's really pissing me off, right? Or you could write the word recessive, right? Because blue eyes are recessive. That's going to be on the phenotype part of the table. Then it's going to ask you for your genotype. So in this hypothetical situation, if you have blue eyes, now, and you don't need to know this yet, but you will after you read the introduction, you will be little b, which means homozygous recessive, right? So if you have the recessive trait, then you're going to be 2 lowercase letters. If, in this case, you have brown eyes, which is a dominant trait, again, on this left-hand side for phenotype, you would write brown or dominant, whichever one you want. And then on the genotype side, you would write big B. Then you would write the word and, because we don't, or excuse me, or, because we don't know what your genotype is. Big B, or big B, little b. That's the first table. You have to fill that out. You have to fill all that out. There's a bunch of traits: eyes, chin color, not chin color, eye color, chin dimple, some things about your fingers, all these things. You need to figure out, write down what your stuff is. And then there's another table after that, and basically you just copy and paste the same information. So the same information that you need to put in for table 1 is the same information you're going to need to put in for table 2. And just to be clear, there's other stuff that needs to be filled in for table 1, but we're going to do that in class. The only thing you need to do before you come to class, before you come to lab, is put your information about your phenotype and your genotype. Any questions about Lab 8? All right. Again, like I've reminded you, exam 1 retake and exam 2 are due next Sunday. That is the end of spring break. If you need help, let me know. If you need more time, let me know. The lab report is due the Monday we get back from spring break. As a reminder, it should have been done this coming Wednesday because the lab report's supposed to be due 2 weeks after the lab's done. But this coming Wednesday is spring break, so I pushed it back to Monday. Midterm grades are due shortly after we get back from spring break. So again, if you're worried about your grades, maybe still spend some time over spring break to fix it. After I turn in the grades, I will take your independent work points and I will add them to your My Online. So for all of you except one person, your grade is going to drop because only one person has finished the assignment. And everyone else, even if you have like 98 points, unfortunately, that's going to make your grade drop because it's not a 100%. Well, I guess that depends on what your actual grade is. Anyway, the point is, for most people, it's going to hurt your grade. So start writing independent work. I'm telling you, I'm begging you, I don't know why you people don't do it. Write me just one paper where you think of a question, answer it, and send it in. Right? I was wondering why my dog's breath stinks. I looked it up, and according to this source, it's because of these bacteria. That's it. That's such an easy paper to write. Just do it at least once so you can learn how I grade. Because I'm telling you, if you wait till the end and you turn a bunch of stuff in and they're all worth 0 from a technicality, that's on you because you never turned anything in to see how I grade. Yes.
It may seem like I know this question, but how many?
Good question. To remind you, you have to hit 100 points. So there is no number. For some people, it might be one paper. They might send in a perfectly written 10-page paper and get the full 100 points. Some people might send, 50 papers, 50 little papers. Just keep turning stuff in until you get 100 points. So in a sense, there's no pressure, because even if your paper sucks, it doesn't matter. Just keep turning things in until you hit 100. It's not like, you have to do it in this many pages. Yeah. So start working on it. I'm telling you, it's really there to help your grade, because a lot of you need some help with the grades, and that's completely in your control. All you have to do is write stuff, and you're in control of what you write about. I'm not going to say, write about something boring, right? Try to find something that you're interested in. Anyway, the last reminder, or actually, this is more of an announcement. The last announcement is to remind you that the last day to drop this class is sometime after spring break. I forget when you should look into it. Some of you should really look into dropping this class. I hate to say that, but unless you can really turn things around, like some of you barely come to class, barely come to lab, and I'm here to help if you need it, but it seems like you don't care. And I get it, you know, life happens. There's other things to worry about. So, but the point is, if you don't care enough because you've got other things going on, save your GPA and try to drop the class, or don't try to do it, just drop it. Or if you want to turn things around, I'm here to help. Anyway, any questions about announcements and reminders? Does anybody have their own announcements? All right, nice and easy. Oh yeah, one more time, just a reminder. I know I say this every time, but so far no one's taken me up on this. Before you turn an assignment in, feel free to give me 5 minutes to meet me online and just say, hey, just can you look this over and tell me if it's going to get any credit, right? Which is big, you know, not like go for nitpicking it. Just say, you know, give me the big stuff. Like, oh man, you completely forgot to do that table. Or whoa, you forgot to do that video. Oh yeah, videos, to remind you. Sorry, this is my last reminder, then I'll move on. When you do the videos and I'm looking for your grade, like I'm looking, like you've turned into pre-lab, I go to the video, I hit Control F, which brings up a little search function, and I'll search like your name or your section number. So if your name's wrong or your section number's wrong, then I'm not going to find it. And no offense, I'm not going to sit around and search for it and go through every single one of the emails of people who've done it. There's been hundreds of people who've done that. So make sure your stuff is right. And once again, you guys who are in this class, you are in 101-02. 101-02. Not 101-03 or 101-01, 101-02. So make sure you get that right. All right. Anyway, now are there any questions or do you have your own announcements? All right, let's get back into the material then. As a reminder, we just finished talking about meiosis 2. We went through meiosis in detail. Now we're kind of taking another step back and looking at the big picture, kind of like a review. So again, we're comparing mitosis and meiosis, except now we're kind of doing it formally and putting all these things in writing. For both mitosis and meiosis, the chromosomes only duplicate once, right? So there's only one chromosome duplication. So it's not like meiosis 1, you go prophase 1, metaphase 1, anaphase 1, telophase 1, and then it goes back into the cell cycle and goes into interphase, right? It's not like that. So we only have the one duplication of chromosomes. The number of divisions, that's where it's different, right? In mitosis, there's only one division, right? So we duplicate once, so we double everything, then we cut it in half, bringing us back down to the original number. And another way of looking at that, if you think about it from the perspective of an exam question, you start with one cell that's diploid, and you end with 2 cells that are diploid for mitosis. In meiosis, again, you start with however much a number of chromosomes, you double it, so now you have twice the amount. Then you cut it in half for the first round of meiosis, so now you're back down to the original number. Then you cut it in half again, so now you have half the number of chromosomes, which is why they are now haploid cells. And again, you started with 1 cell, you cut it in half, now you have 2 cells. Then you cut them in half again, and you end with 4 cells, right? So a lot of test questions on there. You know, which of the 2 produces 4 cells? Meiosis. Which of the 2 produces 2 cells? Mitosis. Which of the 2 produces diploid cells? Mitosis. Which of the 2 produces haploid cells? Meiosis. A lot of test questions there. Any questions about that slide? All right. Before I move forward, let's do another attendance word. The process of meiosis produces haploid cells. How many haploid cells does meiosis produce? That is your next word for attendance. I'm going to move around pretty quickly because that should be pretty simple. So again, this next slide, we're still doing this review where we're comparing mitosis and meiosis because a lot of questions, a lot of the meiosis questions will be based on that, like comparing the 2. The events that are unique to meiosis occur in meiosis 1, not meiosis 2. So again, like I said earlier, in a different way, the way I said it earlier was I said, between meiosis 1 and meiosis 2, which one is more like mitosis? right? So meiosis 2 is more like mitosis. Meiosis 1 is the one that's more unique. Even though it's not on the slide, and I'll remind you, it's because during meiosis 1, that's when we have crossing over. That doesn't happen in meiosis or in mitosis. Meiosis 1, when all those homologous chromosomes line up in the middle of the cell in metaphase, it's random, right? And that's where we get independent assortment. Meiosis 1, since I just said that, we're dealing with homologous chromosomes, not sister chromatids. In mitosis, we're dealing with sister chromatids. In meiosis 2, we're dealing with sister chromatids. It's only in meiosis 1 that we're dealing with homologous chromosomes. And again, like I keep saying, you saw this on the previous slide, here it is written in a different way. Unlike in mitosis, meiosis 2 yields haploid daughter cells. So we go from diploid to haploid. That does not happen in mitosis. All right, any questions about this slide before I show you a visualization of everything we just talked about? Okay, this is great comparing. This picture is not from your textbook. So if you find it helpful for your studying purposes, then you're going to probably want to download this PowerPoint, or at least access it on WVSU Online. It's a Google document, so technically once you open it up, it's there. You don't necessarily need to download it. But anyway, so you have mitosis on the left and meiosis on the right. Both of which, this is start with duplicated chromosomes, right? Here, we're dealing with the sister chromatids only. Here, those homologous chromosomes are together, right? Because it's meiosis 1. Here, the sister chromatids are lined up in the middle of the cell because it's mitosis. Excuse me. Here, in metaphase 1 of meiosis, it's the homologous chromosomes that are lined up in the middle of the cell instead of the sister chromatids. So here, we pull apart those sister chromatids, right, during anaphase and telophase. Here, we pull apart the homologous chromosomes. And then, of course, then it goes through around meiosis 2, which is not shown, right? They just kind of skipped it. But here, meiosis 1, they showed the stages. Meiosis 2, they're just like, yeah, here's the whole thing. And at the end, we end up with 4 genetically unique haploid cells. So any questions about that slide? Okay, so like I mentioned earlier, that independent assortment thing is going to be important later on. And here's where it's important later on. Well, to start with, it's going to come back even after this. But let's talk about the origins of genetic variation. This next bullet point is very important. It's important for when we talk about heredity and genetics. It's going to be important when we talk about evolution, because again, without this genetic variation, evolution would not happen. It would be impossible. You can't, if everything is the same, it can't evolve, you know? Anyway, let's talk about it. The origins of genetic variation. The offspring of sexual reproduction are genetically different from their parents and one another. You already know that. I've already said it. I said it today. I said it previously. I'm saying it again. The offspring of sexually reproduction are genetically unique, right? That's an important thing. So, how is it that meiosis produces this genetic variation? Even though we've already said it, we're going to say it again, perhaps give you some more terms, look at it in more detail, but here we go. Independent assortment of chromosomes. So here we go. We're talking about it again, but now we're digging a little bit deeper. In metaphase 1, as you know, because we talked about it today, the side-by-side orientation of each homologous pair of chromosomes is by chance. So again, it's not like all 23 pairs of your chromosomes line up, where it's mom on one side and dad on the other. It's not like that. It's all completely random. So for any species, not just humans, for any species that has more than 2 pairs of chromosomes, every pair orients independently of all others. So it's the same, basically what I just said. So unless there's an organism out there that only has, 1 pair of chromosomes, then this applies to them. So here's where it gets interesting in my opinion. Humans have 23 pairs of chromosomes. So if you were to do the math, that means it's 2 to the 23rd possibilities of combinations. Meaning, again, mom on one side, dad on the other, right? And then if you could, that's one possibility, is having all 23 pairs of mom on one side, dad on the other. Then the next possibility with all mom on one side and dad on the other, except the last one, which has them switched. And then there's, so on and so forth. Picture that in your brain, all the different ways you can do it. That means there's about 8 million possible combinations. Think about that. So if you were to look, if you're taking, take a microscope and look at 8 million of your sperm or eggs, whatever it may be, that means each one of them would be unique. Every single one of them, statistically, should have a different orientation of which chromosome lined up where. So that's the one part of the statistics. The other part, and you've learned probability, no, I haven't touched the probability yet. Anyway, you'll learn that later. And I'll explain this math later. So there's 8 million possible combinations for a sperm, and also 8 million possible combinations for an egg. And when you combine those possibilities, so that sperm meeting with that egg, there's 64 trillion combinations of chromosomes. This means if your mom and your dad had 64 trillion children, then each one of them statistically would have a different orientation of mom chromosome versus dad chromosome. So just on this is really basic too, it gets a little bit deeper than this, but even just this basic description, again, if your parents had 64 trillion kids, statistically they would all be unique, and you would be different than all of them. Think about that. To me, that's crazy. That means you would not be you if your dad, assuming you were normally fertilized and wasn't IVF or something like that. If your dad didn't ejaculate, when he ejaculated and the way he ejaculated, at that very moment, it wouldn't be you. Think about all the different sperm that could have made it to the egg. So if your dad was a little bit pissed off driving home, and he was a little bit in a bad mood, it took him just 30 seconds longer to get it up. I know this sounds a little bit vulgar, but think about it. So somebody cutting him off could have made the difference between you and some other version of you. You or you, like it has to be that exact thing. It's so crazy to think about. And also think about too, your dad had to have that exact moment for him to be him, and your mom her. And then obviously if they weren't them, then it's just mind blowing. Anyway, you might need to know that word for the exam. Not the word, excuse me, these numbers. So for humans, there's 8 million possible combinations of chromosome alignment per gamete. In other words, 64 trillion combinations when you're talking about fertilization. And keep in mind too, the only thing I'm talking about so far is how your chromosomes are lined up. Whether you got, for those 23 chromosomes, whether you got your mom's version or your dad's version. I haven't even talked about crossing over. So even with the 64 trillion, even if somehow your parents did have 64 trillion children, then, or even double that, then you have to take into account, you might have a mom chromosome, but it might have a dad chunk on it, or it might not, or it might have 2 dad chunks on it, right? So we talked about crossing over. So all these numbers are actually small. 64 trillion is small compared to the reality. In reality, it's much larger because of crossing over. But anyway, here's a visualization of what we're talking about. This is independent assortment, and this is only looking at a species that has 2 pairs of chromosomes. We have 23 pairs. So here, the first possibility is you got all mom on one side, all dad on the other. Over here, we've got the opposite, right? Mom on one side, dad on the other for chromosome number 1. And for chromosome number 2, we have dad on one side, mom on the other, right? So with that possibility, you make these many eggs, these gametes, whether they be egg or sperm, or this possibility, you make those. See, each one of those is unique. So for this species that only has 2 pairs of chromosomes, there are 1, 2, 3, 4, 5, 6, 7, 8 different possibilities of gametes. And again, that's only 2 pairs of chromosomes. Imagine trying to draw that for humans. That would be an independent work project. You literally wouldn't have enough time in a year to draw all the different possible combinations if you were to do a human chromosomes. Anyway, any questions about this slide? All right. So again, that was independent assortment. We just kind of looked at it a little bit deeper. And unfortunately, we're going to look at, I say unfortunately, but we're going to look at independent assortment again from a different context. At that point, it'll actually, in my opinion, get a little bit more complicated. But for now, we'll leave it at that. Then there's crossing over, which is what we've already talked about, right? Crossing over is the exchange of corresponding segments between homologous chromosomes. And I'll show you a picture of it on the next slide. But again, you got your mom chromosome, your dad chromosome. They're homologous chromosomes, so it's the same one. Like maybe chromosome number 13 or whatever. You got one version from your mom, one from your dad. They are in a synapsis. Remember, that's the word I used earlier. They're together because it's prophase 1. So they're kind of playing footsie, if you will. They're in really close proximity. So what happens? Sometimes they swap chunks. As I've shown you before, here's another picture of it, right? Here they are, close together, and you can see they're interacting with each other, and then boom, they swap chunks. Of course, this is just one of the ways it can happen. So in this case, they did swapping twice, right? So there's 2 chunks that were swapped. Sometimes it's just 1 chunk. Sometimes it doesn't happen at all. Sometimes the chunks are big. Sometimes the chunks are small. So there's all these different combinations. And again, think about this, and then add that to that whole idea of 64 trillion possible combinations of egg and sperm. This is, again, the numbers are just mind-blowing. And again, I say, you would never be you if every little tiny thing didn't happen the way it did. Of course, I guess that might be a little slightly different with IVF, but that's a whole different conversation. Anyway, any questions about crossing over? All right, let's jump into this. This will be good. We might be able to finish it if I go quick enough. As far as the exam is concerned, this next section is not too important. The concepts are very simple to understand. So let's jump into it. And you've already kind of been familiarized with this in the last portion of this week's lab. All right, so let's talk about what happens when there's mistakes, right? What happens when we have errors in meiosis? This can result in genetic abnormalities ranging from mild to severe to fatal. We're going to talk about them. Obviously, they're visible with a karyotype. That's what you did in lab, right? You can look at all the chromosomes and go, oh, that's an extra chromosome number 13. Or, oh, man, that's an extra chromosome number 18. Oh, speaking of which, really quickly, so one person got this wrong, and it's not past due yet, so you can still fix it. When you look at those karyotypes and you look at how, like, oh, that's an extra chromosome number 13, then you go to that chart and say, oh, that's, I think, Patau syndrome. I forgot the name of it. Doesn't matter. Once you know the name of the syndrome, then you're supposed to go, like, Google it, use the internet, look it up, and then write down what the internet says. Like, oh, if you have trisomy 21, this is what your kids can expect. You know, like, that's what you're supposed to do. So at least one person hadn't done that yet. So just let you know that's what I expected of you. So if you missed those points, that's why. Anyway, let's talk about it. How does this happen? Long story short, it's something called non-disjunction. You need to know this. Non-disjunction. And I like that. It's one of those biological terms that actually makes sense, kind of describes the situation. Non-disjunction, when they don't separate, right? Chromosome pairs fail to separate during anaphase. As we're going to learn later, this can happen in meiosis 1 or meiosis 2, meaning it could be the homologous chromosomes that don't separate, if it's meiosis 1, or it could be the sister chromatids that don't separate, if it's meiosis 2. Either way, the end product is the same, right? No matter what. If you have non-disjunction, what you're going to end up with are gametes, or egg or sperm, that have an abnormal number of chromosomes. Maybe they have too many, maybe they have too many, maybe they don't have enough. Any questions about this slide before we go to the next slide and talk about this a little bit more? Okay. If this doesn't happen, right? So when a mistake doesn't happen, then we describe these cells as euploid. So just another word for you to remember, right? Euploid means like the true number, right? That when there were no mistakes, it has the right number of chromosomes, that is euploid. When there's a mistake and there's too many chromosomes or not enough chromosomes, we call that aneuploid. So the not true number. And there's the way we're going to subdivide it, there are some other subdivisions, but the ones we're going to talk about, aneuploid, there's 2 different types of aneuploidy that we talk about. There's monosomy, or monosomy, depending how you say it, and trisomy. And again, this is kind of in the name, right? Mono means 1. So if you only have 1 chromosome, but you're supposed to have 2, it means obviously you don't have enough, right? If you have trisomy, try 3. If you have 3 chromosomes, but you're only supposed to have 2, obviously you have an extra. For independent work, you can look up tetrasomy. So there's also, and again, you don't need to know it for the exam, but there's also, it gets bigger than this. Sometimes there's more than 3, sometimes there's 4, and so on and so forth. You can look this up and look into the details, but generally speaking, for humans, it's really bad. For plants, it can be good. Sometimes in agriculture, we actually take advantage of that. Like having double the number, which would be, beyond trisomy, double the number of chromosomes can sometimes be good in plants and agriculture. You can look that up if you want. Anyway, here's a picture of what we're talking about. Here's some non-disjunction. Here on the left-hand side, the non-disjunction is happening in meiosis 1, meaning it's the homologous chromosomes that didn't separate, obviously, because it's meiosis 1. On the other hand, on this right-hand side, the non-disjunction, meiosis 1 went fine, no problem. Here, in this example, meiosis 2 had the problem. So it's the sister chromatids that failed to separate. Either way, the result is, I don't want to say the same, it is different individually, but the same as in your gametes are going to be screwed up, some of them, right? So this one has too many, this one has too many, this one doesn't have enough, this one doesn't have enough. This one has too many, this one doesn't have enough. This one's normal, and this one's normal. So obviously, one is worse than the other, and that'll be the next word for attendance. Which one is worse? Non-disjunction 1, non-disjunction of meiosis 1, which is on the left-hand side, or non-disjunction in meiosis 2 on the right-hand side. The hint is to look at the products. What are the products? What are the results of non-disjunction of meiosis 1 versus the products of non-disjunction 2? Which one is worse? Not that I'm necessarily going to ask you that for the exam. Once again, this is just attendance word, right? Just to make sure you're paying attention. What I haven't told you, and you could look this up for independent work, is what's causing this? Like, why in the world are they not separating? So you can see, for example, here, the mitotic spindle is attached here and here, but there isn't one here. Is that all it is? Was it just the fact that the mitotic spindle failed to attach there? Or does it attach and accidentally break off? These are things you don't even know for the exam, but you can look into it for independent work. Like, what causes non-disjunction? Also, are there environmental factors that cause it? Like, are there certain drugs that a mom could take or a dad could take that would increase this happening? Is there a certain age that the parents could be that would increase the probability of this happening? All things you could look up for independent work. So anyway, those are our abnormal gametes, right? This is how we produce abnormal gametes, so these different types of non-disjunction. The next slide I'm going to show you is going to be even more simple than that. It's just going to be a picture of an abnormal gamete combining with a normal gamete. And yeah, there's a lot of details. Don't worry about it. This is just an example. So here we have a normal sperm cell combining with an abnormal egg. Right, so the sperm again has two pair of chromosomes. Two chromosomes. And the abnormal, the female, the egg, has an additional one, whatever chromosome, that bigger one, it has an additional one of those. So once it combines, now we have a zygote that has one too many chromosomes. Any questions about that? All right, very quickly, we might be able to finish this off. The most common one is Down syndrome. I'm not going to ask you, I don't think I'm going to ask you any questions about Down syndrome, but the reason I'm bringing it up is because it's in the textbook, and the reason it's in the textbook is because it's the most common of these abnormalities. You know what? There's 1 minute left, so this will be interesting. When we get back, so we're done learning about all like how non-disjunction works. So when we get back, I'll give you some examples, like trisomy 21, of the results of this non-disjunction, which is unimportant for the exam. And then we can just jump right into the next chapter. So the last question will be, of all the chromosome abnormalities, let's not even do one more question. So the last thing you need to do for attendance, as usual, is send me one question about the material that I covered. For grading purposes. Obviously, you can send me a million questions, but for grading purposes, make sure the question is about these materials. This material. That's it. I'll be in my office and online at least until noon for office hours, and I'm going to go home and I'll be back online working from home. So if you need me, let me know. Have a good spring break. I will be working for spring break, so I'm there if you need me.